I'm going to draw for you retroviral genome here. And this is the retroviral genome after reverse transcriptase has converted the retroviral RNA into double-stranded DNA. And the retroviral genome has a long terminal repeat of the same sequence at both ends. And for the purposes of this discussion, this will be the HIV genome. Because of its medical importance. And we will have a little blow-up of the HIV genome. And I'll just draw a few nucleotides here for illustration. That's the upper strand, and here's the lower strand. And these two nucleotides right here are critical for what's going to happen next, because it's going to turn out that the retroviral integrase will remove two nucleotides from the 3' ends, both 3' ends. So I'll remove two of the nucleotides from this particular end, leaving us with a recessed 3' hydroxyl group. Now I'm going to draw the target of the retroviral integration event, and that'll be the host genome. The retrovirus wants to jump into the host genome, so we will draw the host genome for you. Double-stranded DNA there. And I'm going to show you the retroviral genome curled up. And it's curled up because the retroviral integrase will hold on to both LTRs. So we've got the two unpaired nucleotides at the 5' end of the upper strand on the previous diagram. Ending in a 3' hydroxyl here. And then two unpaired nucleotides of 5' end of the other strand, the lower strand, on the previous diagram, ending in a 3' hydroxyl there. And these 3' hydroxyls will be used by the integrase to catalyze nucleophilic attacks on the host DNA. So we will draw that. And the nucleophilic attacks occur offset from each other. And the offset here I'm going to put in black brackets. And those that bracketed sequence between the two sites of nucleophilic attack is going to be destined to become a short direct repeat. And you'll see that by the time we're finished. So, just to review, the integrase catalyzes nucleophilic attacks on the host DNA, both strands of the host DNA. And I'm going to show you the results of those nucleophilic attacks. So starting, and this time I'm going to start with the lower, I'll draw the diagram from the lower strand, the 3' end of the host DNA, moving over to that site. And then that nucleophilic attack, actually creates a covalent bond with the retroviral DNA. Here's the 5' end. And now, if I draw in, let me finish drawing the bottom strand here. Here's the end of the bottom strand, 5' end. Now, starting from the 3' end of the upper strand, we'll show you the result of the nucleophilic attack. On the upper strand. And the lower strand, the upper strand, I'm sorry, let's draw it in blue. Comes all the way up to there. There we go. So this configuration is a little hard to see. So what we need to do is to pull the DNA ends out so you can see everything. I'm not going to change anything. All the bonds are still there. It's just the geometry will change for ease of viewing. So again, if I start with the 3' end of the lower strand, of course we want to keep our color coding. So if we're talking about the 3' end of the lower strand, we want that host DNA to be blue. It's going to be covalent linked to the retroviral DNA. There's the retroviral DNA, and it's protruding 5' end. The same thing goes, and then the continuation, of the host DNA over here. I'm leaving a gap there on purpose. You're going to see why I have to do that. Because if I start drawing the 5' end of the retroviral upper DNA strand, that only goes up to this point. Let me draw it a little more carefully. It only goes up to that point because it doesn't reach all the way over because there's a gap that's exactly the distance that between the two nucleophilic attacks on the upper part of the diagram. And if I continue drawing the retroviral DNA, I come up to here. Let me curl this thing down to make it a little easier for you to see. Okay. And then we have the point of covalent attachment there. And voila. We've got another gap over here. So that is the previous diagram stretched out. Nothing has changed. What happens next is that nuclease comes along and removes the protruding 2 nucleotides at the 5' ends, the unpaired 2 nucleotides. And then host polymerase, we'll draw that in blue, host polymerase, DNA polymerase, and ligase repair the gaps. Okay, now I'll draw that Here's the polymerase coming along, and then the ligase finishing the job. Polymerase coming along, ligase finishing the job. And voila, you've got the retroviral DNA integrated into the host genome. And the epilogue is, of course, understanding retroviral integration has led to the development of HIV inhibitors, which are now being used to treat patients with HIV infection. Thank you so much.